In this video, I will teach you how to set temperature or membrane fluctuation parameter individually for each cell. So in the previous video, we have added new cell type, non-condensing. And we have not seen any cells of type non-condensing so far because we are starting with a single cell that we create in this steppable in start function. This is our first cell. Then this cell is growing, and then as it grows, it gets divided into daughter cells, and the daughter cells keep growing, and so on. But nowhere in our code, we set cell type to be 2, right? We have only cell type 1, cells of type 1, which is condensing. So let's change it. So in our update attribute functions, let me actually explain what is going on in update attribute functions. I will get rid of some of the commented out code to make things cleaner. So let me actually walk you step by step what's going on inside this steppable. So at each Monte Carlo step, we create a list of cells that will get divided. So this is an empty list of cells. Next, we go through every single cell that we have in our simulation and we check if the volume of this cell is greater than 50. If it is, we append this cell whose volume is greater than 50 to the list of cells to divide. And then there is another loop where we iterate over the list of cells to divide, so we iterate over this container, and we perform the division. And we divide cells along randomly oriented cleavage plane or cleavage line. The moment that we call this function, right after the division, that creation of new child cell takes place, the function called update attributes gets called. Okay? And in this update attribute function, first thing that we do, we reset target volume of the parent cell to be half of what it was before. Okay? Next, we clone all the attributes of the parent cell and assign them to the child cell. So the new cell that gets created in this function does not sets cell attributes to the default values, and that's not what we want. We essentially want to initialize them to some sensible values. And this function, clone parent to child, performs those tasks. And finally, in those if-else statement, we can do some simple type differentiation. So for example, if parent is of cell type 1, we can change the child cell to be of cell type 2. Or we can type self non-condensing. Okay? We can replace this statement with self and let's run this code. And then I'll show you one extra step that we can perform, and then this will be the end of our lesson. So this code is running, and as a result, we should see cells of different cell types getting created. That's what we want. Now, what I would like to do is that anytime we create a cell of type non-condensing, I want this cell to have more jagged boundaries. So in other words, I want the membrane of the cell to fluctuate more. So how do I do this? Well, I can go to CC3 Python menu, go to cell attributes, and select fluctuation amplitude. And this select paste code snippet that provides me a starting point for my modification. So essentially what I'm going to do is I'm going to modify this code so that it corresponds to what we actually have. So after we create a cell and we assign its type to be non-condensing, I'm going to also increase the fluctuation amplitude by 100 units. So that's a lot. Why is it a lot? That's a lot because in our simulation, this fluctuation amplitude, also referred to as temperature in POTS terminology, has value of 10. And the reason I'm setting it to 100 is so that we can see on the screen what happens. So essentially what I'm trying to teach you here is how to customize or how to customize behavior of cells right after you do mitosis. Okay, so let me stop the simulation. Let me make sure that all my files are saved. They are, because we see the blue floppy disk symbol. And that's what happens. It's not super visible, but you can see that those green cells have more jagged boundaries than the blue ones. And that's precisely what I wanted to achieve. All right, so this is it for this lesson, and I'll see you in the next video.